Hello, good evening, guys. So, I'll start today's session. I'll share my screen, and one of you can let me know if my screen is visible and if I'm audible. Okay, so yes, good evening, guys. Today is the ninth live session of this series of live sessions that we are having. So, in this live session, what we do basically is we discuss questions and concepts behind the various topics in this course genetic engineering theory and application taken by Professor Vishal Trivedi from IIT Guwahati. And in these live sessions, what we do is that basically uh, check some problems and we try to figure out the answers to those problems, some old assignment questions, and we also discuss the concepts behind those questions so that we also have a better grasp on the element and so that it helps us in answering similar type of other questions. So can one of you unmute and let me know if my screen is visible and if I'm audible?
Yes, ma'am.
Okay,
Your screen is visible
okay,
and it is
okay.
on.
Okay, thank you so much. So we'll move on to the first question for today. Okay, so the first question for today is, in the turbidimetric method, the OD of the bacterial culture is measured at 220 nanometer, 400 nanometer, 500 nanometer, and 600 nanometer. Okay, so can one of you, I'll wait for 2-3 minutes. Can one of you try answering this question and with the reason why you're choosing that answer as well? Okay, so the correct answer is 600 nanometer. Amongst the option given here, it's 600 nanometer. Some people take OD measurement even at 590 nanometer, but basically 600 590 nanometers as the uh, wavelength. Basically we choose to take the absorbance or optical density when we are trying to find the bacterial growth. Basically when we are trying to follow the bacterial growth curve that we discussed in one of our previous classes. So that is what happens in case of turbidimetric method. So if we talk a little bit in detail, so what is turbidimetry? It's basically a simple rapid method for following growth. So spectrophotometer or calorimeter can be used for the turbidimetric measurements of cell mass, and a spectrophotometer is used to determine turbidity, which is basically cloudiness, by measuring the amount of light that passes through suspension of cells. So more cells will mean more turbidity, and more turbidity, that means less light is passing through the suspension. Okay, so basically what you do in absorbance, you basically put a solution and you want to figure out the transmittance, like which you pass a light and what amount of light is getting transmitted. Anything that is not getting transmitted but was incident is basically being absorbed. So when we are using this technique for doing turbidity experiments, so we basically try to figure out the cloudiness or the turbidity, because if there is lesser number of bacteria, the solution will be less cloudy or less turbid. If there's more number of bacteria, the solution will be more turbid, right? So using spectrophotometer, we basically measure the amount of light that passes through the suspension of cells. So when there are less cells, more amount of light will pass through. That means the transmittance will be more, the OD will be less. But if there are more cells, there will be more turbidity. So the transmittance, like the light passing able to pass through, will be less. That means the OD or the absorbance will be more. Okay. The culture should be dense enough to register some amount of turbidity on the instrument, right? It may not be possible to measure cultures grown in deeply colored media or cultures that contain suspended material other than bacteria, because that will give you false data, because if it's a deeply colored media and if your media component which is giving it the color, if that component has any kind of contribution to the absorbance at that particular wavelength, then that is an issue. And also if there is other particle in your solution except bacteria, for example dust particle or anything else, that will also lead to scattering of light, and that will also give you a false data. And also it must be recognized that dead as well as living cells contribute to turbidity. So when we are checking turbidity, there might be some cells, bacterial cells, that are dead, right? And there might be some cells which are alive, but both living and dead cells will make the solution turbid. So just by measuring OD, you cannot say that this compare it. You cannot any do it, even do any comparative analysis that this amount or this percentage of bacteria is alive or dead, because both alive and dead bacteria will contribute to the turbidity. Okay. Now what we'll do, we basically calculate the percent transmission. Okay. So if there are fewer cells present, there is less turbidity and this will allow more light to pass through, as I explained in the previous slide. So the percentage transmission is higher when the cell number is lower. Okay, but the absorbance of the OD is lower. Absorbance is the opposite of percentage transmittance. So more light is absorbed when more cells are present, and this absorbance goes up as turbidity goes up. Okay, basically it's not absorbance in this case, it's scattering. When light is scattered, it will not be transmitted, right? It is scattered in different directions. Okay, so if there are more number of cells, if it is a dense turbid solution, light will be scattered. And if light is getting scattered, that means less amount of light is passing through. So the percentage transmittance is less. That automatically means that the percentage that was absorbed or scattered was more. So this is what I'm trying to say. This is schematic representation. This is a light source, this is your cell. So if your cell is if this is your solution, if your solution is clear and not dense, then the entire light will pass through, right? So this is a light sensitive detector. So you can see 100% light that was incident is transmitted. Okay, but then if you have a bacterial suspension in this tube and your incident light encounters these bacterial cells, the light will be scattered. If you see, these are scattered light. These will not reach the detector. So only a little amount of light will reach the detector, and so your percentage of light transmitted will be much lower, right? Hmm. Now, another indirect way of estimating bacterial numbers is measuring the metabolic activity of the population. For example, if you want to figure out the percentage of live bacteria, the best way will be to figure out the metabolic activity of the bacteria, because only live bacteria will contribute to the metabolic activity. For example, acid production or oxygen consumption, nutrient utilization, waste production, pH, etc. Now, the assumption is that the amount of acid produced or oxygen consumed under specific conditions and during a fixed period of time is proportional to the magnitude of the bacterial population. Admittedly, the measurement of acid or any other end product is a very indirect approach to the measurement of growth and is applicable only in special circumstances. Right? If we talk about dry weight of pre-weighed filter paper containing pellets of microbial cells is measured. And dry weight of filter paper is nullified by subtracting the dry weight of only filter paper of similar size. Thus, the dry weight of microbial cells can be obtained. However, it can be used only when very dense suspensions and the cells must be washed free of all extraneous matter. So, moreover, dry weight may not always be an indicative of the amount of living material in the cells. So, dry weight may continue to increase without corresponding cell growth due to the accumulation of intracellular reserve materials. Yet, for many organisms, the determination of dry weight is an accurate and reliable way to measure growth and is widely used in research. So, what are the advantages? Only way to determine growth of filamentous bacteria, it is rapid and easy. Disadvantage, it's cumbersome, not very accurate. It does not give you cell number or increase in mass. It cannot distinguish between live and dead cells and must work within certain absorbance. So, in brief, there are some of the indirect methods of measuring microbial growth. It helps us to understand that it is not always necessary to count microbial cells to estimate their numbers. So, in science and industry, microbial numbers and activity are determined by some of the indirect methods as well. So, spectrophotometer is used to determine turbidity by measuring the amount of light that passes through a suspension of cells. And an indirect method of estimating bacterial numbers is measuring the metabolic activity of the population. So, for filamentous organisms such as fungi and molds, measuring dry weight is a convenient method of growth measurement. Now, what is the scintillation counter? So, even we can do it via scintillation counter as well. We don't need to get into the details of this mechanism, but anyway, briefly. So, the radioisotopes, apart from those causing ionization radiation, are able to emit photons of light that cause excitation. So, this excitation can be detected and quantified. So the detection and measurement of this excitation is known as scintillation, and the instrument which detects the light using a photomultiplier is known as scintillation counter. So the electron pulse which is detected by the multiplier is a result of conversion of light into electrical energy, right? So this is how the scintillation counter works. I don't know how many labs basically use it. Mostly it's the OD method only. So we'll not go into much of the scintillation dynamics. Okay, we'll not go into this. So coming back to the question that we started with basically. So in the turbidity method, the OD of the bacterial culture will be measured at 600 nanometer, right? So basically bacterial optical density is measured at 550 or 600 nanometer because at these wavelengths the light scattered caused by bacterial cells in suspension is prominent without significant interference from other compounds that might also absorb light. Okay, so the cells themselves do not absorb much at these wavelengths, allowing for an accurate measurement of cell concentration based on light scattering. Okay, so bacterial cells are typically in the micrometer range, right? And light in the visible spectrum around 5 to 600 nanometer scatters well of these cells of this size. The amount of scattered light increases with cell concentration. So OD provides an indirect measure of cell density. And the 550 nanometer or 600 nanometer wavelengths avoid absorption from many common media components, proteins or metabolites that might be present in the culture. So these wavelengths allow the OD measurement to reflect primarily the turbidity or the scatter rather than absorption, making it a very good proxy for bacterial growth. So OD600 has become a standard in microbiology, allowing easy comparison across experiments and studies. So OD550 is less commonly used because it can be applied in certain cases where a slightly different wavelength might reduce interference from other substances in the media. So in essence, 600 nanometer is the sweet spot basically for measuring bacterial growth because it minimizes background noise from non-cellular materials and maximizes sensitivity to bacterial turbidity. Okay, I hope you understood. If you have any questions, you can ask me now. I'll wait for 5 minutes. Okay, so we'll move on to the next question for today. Yeah. So the aqueous phase in phenol chloroform extraction contains the proteins, DNA, lipids, or polysaccharides. I'll give you a few minutes if you can figure out, then we'll discuss.
And the answer is option B,
Okay.
DNA.
Okay. DNA. Okay. So you're right. The aqueous phase is option B, DNA. So we'll discuss a little bit about phenol chloroform extraction. So if you talk about DNA extraction, it's basically a fundamental technique in molecular biology and genetics that involves the isolation of DNA from eukaryotic or even prokaryotic cells or tissues for various downstream applications, right? Such as DNA sequencing, PCR, genetic testing, molecular research, etc. etc. So it's a crucial step in many scientific and diagnostic procedures, and several methods are available for this purpose as well. So you can imagine the start of molecular biology is basically all these um, you know basic techniques, DNA RNA extractions, PCR, and all of these are not basic, these are fundamental, that's the right word. These fundamental techniques make up molecular biology. So there are various techniques that have been optimized till date, and chloroform extraction being one of the prominent ones. So what basically, in order to come up with the technique, you need to have a prior um, objective, right? A step down, clear cut objective. What do you want to do? So, first, in order to take this DNA out, you have to disrupt the cells. So, the first step in DNA extraction will be to break open the will be to basically break open the sorry break open the cells or tissues containing the DNA, and this can be done using various methods such as mechanical disruption, which can be done by the grinding or homogenization, then chemical lysis using detergents or enzymatic digestion. Then you have to do protein removal. So after cell disruption, the DNA is typically mixed with proteinase K or other proteolytic enzymes to degrade the proteins and to remove them from the mixture. Now, proteins can interfere with downstream DNA analysis. So there are protocols where you have to isolate the protein. There are protocols where you have to isolate the RNA. There are protocols where you have to isolate the DNA. But if your objective is to isolate the DNA and do downstream work, further analysis, then you have to make sure that you separate the proteins. Because, for example, if you think you have isolated the DNA and if there are any protein components, those will interfere with your next step analysis. So that's why you should always remove the proteins. Then DNA purification. The next step involves separation of DNA from other cellular components like RNA, proteins, and cell debris. And this is typically achieved through a combination of centrifugation, precipitation, or filtration methods. Now, DNA precipitation is DNA is often isolated by adding a salt solution, for example, sodium acetate and all of that, and cold alcohol like ethanol or isopropanol to the DNA solution. So this causes the DNA to precipitate out of the solution as long stringy strands. Then we'll do the washing and resuspension. The DNA precipitate is then washed to remove any remaining contaminants and salts, and it is then resuspended in a buffer solution suitable for the intended downstream applications. So what are the common methods for DNA extraction, right? So the common method for DNA extraction, one of that is phenol chloroform extraction. So this method involves the use of phenol and chloroform to separate DNA from cellular components. It yields high quality DNA. Or people do salting in methods. So this method will use a high concentration of salt like ammonium acetate to precipitate DNA. It's a relatively simple and widely used technique. People do silica based DNA extraction with spin columns. So in this method, DNA binds to the silica membranes in the presence of chaotropic salts, and after several washing steps, the purified DNA will be eluted with a low salt buffer, right? So spin columns are commonly used for plasmid DNA purification, right? Or small scale DNA extraction. So what are cheotropic salts? Basically, cheotropic salts are substances that disrupt the hydrogen bonding network between water molecules and destabilize proteins. Okay, so how do they do this? They do this by altering hydrophobic interactions. Like cheotropic salts can reduce the hydrophobic effect, which prevents nonpolar groups in proteins from interacting in aqueous solutions. Or they can unfold proteins and increase the solubility of hydrophobes, or they can destabilize hydrophobic aggregates. Basically, cheotropic salts can destabilize the hydrophobic aggregate solution. So, for example, guanidinium thiocyanate. Thiocyanate is a common cheotropic salt used in solid phase extraction, and it's a potent cheotropic salt that disturbs the structural activity between water and macromolecules like proteins and nucleic acids. Okay, or people do calyx resin extraction. So this method relies on using a chelating resin, right, to bind divalent metal ions like calcium and magnesium and all of that, and basically facilitate the release of DNA from cells. It's a quick and easy technique often used for PCR-based applications. Then we have magnetic bead based extractions. Magnetic beads coated with specific DNA binding molecules are used to selectively capture and purify DNA. So this method is automation friendly and suitable for high throughput applications. Right. And then uh, there's organic solvent extraction, CTAB. It is basically cetyl trimethyl ammonium bromide, and it is used to lyse cells and separate DNA from other components. So this CTAB is a pretty strong detergent, which is why it is commonly used for plant DNA extraction. Okay, so CTAB is a cationic detergent, and it disrupts membranes because it interacts with biological membranes, which helps release the nucleus and sequesters lipids. It binds to polysaccharides and polyphenols, which can interfere with DNA isolation, and it can be used in combination with other compounds such as PVP or ADTM to improve the extraction process. Okay, so that is CTAB is mostly used for uh, this only, like a plant DNA extraction only. Sorry. Okay. Next, so this is the phenylchloroform extraction schematic. If you see, these are the cells, then add STS and proteinase K. This is the cell lysate. You will incubate it, and then if you add phenylchloroform, the lipids will go down, and vortex and centrifuge, basically lipids and proteins will go down, and DNA will be in the aqueous phase. So then you transfer and retain the aqueous phase DNA. So this is silica based DNA extraction. There will be silica resins over here, and the DNA will go and attach to it, and then you can use a low salt buffer to elute the DNA. Calyx resin extraction is like this: cells, you add water, add calyx resin, then you boil and centrifuge, and then you transfer and retain the DNA extract. Or you have magnetic bead based extraction, so sample plus magnetic beads, you mix and incubate, then you separate, basically wash, elute, and then you get purified DNA. So what you do is, basically this is a crude sample containing DNA. You add silica magnesium beads to lysed cells, and silica magnetic beads to lyse cells and tissues. Then use a magnetic rack. You apply the magnet to isolate the DNA bound to beads. Then you wash DNA bound to beads, and you leave DNA from beads. Now for bacterial DNA extraction, it involves first lysing the bacterial cells typically with a detergent or enzymatic solution to release the genomic DNA, right? So even if for bacterial, genomic DNA will be much more longer in size and much more complex than plasmid DNA, right? Plasmid is an extra chromosomal circular DNA. So how do you separate genomic DNA from plasmid DNA? That is also a complex process. So following cell lysis, proteins and cellular debris are removed through a series of purification steps, often involving phenylchloroform extraction or spin column based methods. Now the resulting DNA can be further purified and concentrated, making it suitable for various applications like PCR, DNA sequencing, or genetic analysis of bacterial genomes. So, this process is crucial in molecular biology and microbiology research, enabling the study of bacterial genetics and gene expression. If we talk about the procedure, this is a brief schematic of the procedure. So, schematic brief flowchart or step by step of the procedure. So, you add overnight culture to tubes and centrifuge it to get the cell pellets. Okay, you remove the supernatant, it is nothing but media, used media. Then you add lysis solution, then you incubate the samples for some time. Then you do the protein precipitation step, then centrifuge for some minutes. Then you do the DNA precipitation steps, and you open off tube containing isopropanol and centrifuge. Then remove the supernatant, then you add ethanol and centrifuge again. Then you watch not to lose the DNA pellet, invert the tube and allow to dry on absorbent paper for 10 minutes and all of that. Then you add 50 microliter RNAs and incubate for 30 minutes at 37 degrees Celsius. You repeat that step. Then there's hydration of DNA, which you add some amount of DNA hydration solution, resuspend by micropipette the white pellet, and then incubation at 10 degrees Celsius, which will aid in the dissolution of the DNA. Then you store at minus 20 degrees Celsius. So this is basically how people recover DNA from a variety of sources for biological evidence, like in forensic studies. So what are the essential components? It's basically maximize DNA recovery, remove any kind of inhibitors, remove or inhibit nucleases which will otherwise cleave DNA, maximize the quality of DNA. So how much DNA can be recovered? A diploid cell will contain approximately 6 picogram of DNA. Like for example, sperm cells contain approximately 3 picogram of DNA. So the average white blood cell of adults will have this many cells per ml of blood. So therefore, the theoretical recovery of DNA per microliter of blood should be 30 to 60 nanogram. Okay. So this is a little bit information about PCR reactions leading the number of amount of DNA. So the common method is basically organic phenylchloroform extraction or non-organic proteinase K and salting out. So we'll focus mostly on the organic extraction. So it's most basic of all procedures in forensic molecular biology and the purification of DNA. So the key step will be removal of proteins. It can be often be carried out simply by extracting aqueous solutions of nucleic acids with phenol or chloroform. So it's a cell lysis buffer that will lyse cell membrane, nuclei are intact, pellet nuclei. Then it will resuspend nuclei and sodium chloride sulfate or STS, which is a detergent. Proteinase K, it will lyse the nuclear membrane and digest the protein. Now DNA will be released into the solution, will be extracted with phenylchloroform to remove the proteinaceous material. So DNA will be precipitated from the aqueous layer by the addition of ice cold 95% ethanol and salt, and the precipitated DNA will be washed with 70% ethanol, dried under vacuum and resuspended in tris EDTA buffer. Okay. If we talk about the reagents, what are cell lysis buffers? Which buffer can break the cells? So it will have a non-ionic detergent, salt buffer, EDTA is designed to lyse outer cell membrane of blood and epithelial cells, but will not break down nuclear membrane. Okay, it's a chelating agent of divalent cations such as Mg, magnesium. Now, Mg2 plus is a cofactor for DNase nucleases. If the magnesium is bound up by EDTA, nucleases are inactivated. Then proteinase K is usual to remove most of the protein by digesting with proteolytic enzymes such as pronase or proteinase K, which are active against a broad spectrum of native proteins before extracting with organic solvents. So proteinase K is approximately 10-fold more active on denatured protein. Proteins can be denatured by STS or by heat. So then we talk about phenol chloroform. The standard way to remove proteins from nucleic acid solution is to extract once with phenol, once with the 1:1 mixture of phenol and chloroform, and once with chloroform. So this procedure takes advantage of the fact that deep proteinization is more efficient when two different organic solvents are used instead of one. Also, the final extraction with chloroform removes any left traces of phenol from the nucleic acid preparation. So, phenol is highly corrosive and can cause severe burns. Phenol often means phenol equilibrated with buffers such as TCDTA and containing 0.1% hydroxyquinoline and 2.2% beta-mercaptolithanol, which are added as antioxidants. So the hydroxyquinoline also gives phenol a yellow color, making it easier to identify the phases as well, right? Now chloroform, which often means a 24:1 volume by volume mixture of chloroform and isomyl alcohol. The isomyl alcohol is added to help prevent foaming. And the phenol chloroform isomyl alcohol ratio will be 25, 24 and 1. So the most widely used method for concentrating DNA is precipitation with ethanol. So the precipitate of nucleic acid forms in the presence of moderate concentrations of monovalent cations, is recovered by centrifugation and re-dissolved in an appropriate buffer such as TCDTA. And the technique is rapid and is quantitative even with nanogram amounts of DNA. So the four critical variables are the purity of the DNA, its molecular weight, its concentration, and the speed at which it is pelleted. So DNA at concentrations as low as 20 nanogram per ml will form a precipitate that can be quantitatively recovered. Typically, 2 volumes of the ice cold ethanol are added to precipitate the DNA. So very short DNA molecules less than 200 bp are precipitated inefficiently by ethanol. So the optimum pelleting conditions depend on the DNA concentration. Relatively vigorous microcentrifuge steps such as 15 minutes at or below room temperature at 12,000 rpm are designed to minimize the loss of DNA from samples with yields in the range of a few micrograms or less. Now, solutes that may be trapped in the precipitate may be removed by washing the DNA pellet with the solution of 70% ethanol. So, to make certain that no DNA is lost during washing, you can add 70% ethanol until the tube is 2/3 full. You can vortex briefly and centrifuge. So, after 70% ethanol wash, the pellet does not adhere tightly to the wall of the tube. So great care must be taken when removing the supernatant, right? Because you should not contaminate it with the pellet. Then isopropanol may be used in place of ethanol to precipitate the DNA, and precipitation with isopropanol has the advantage that the volume of liquid to be centrifuged is smaller. So isopropanol is less volatile than ethanol and it is more difficult to remove the last traces. Moreover, solutes such as sodium chloride are more easily co-precipitated with DNA when isopropanol is used. Now, what do we use to resuspend and store the DNA? TE buffer, it is basically tris EDTA buffer. Okay. Tris is basically a buffering agent, EDTA is the chelating agent. DNA is suspended and stored in the T buffer. DNA must be stored in a slightly basic buffer to prevent depurination, and the EDTA chelates any Mg2 plus helping to inactivate DNases, right? Then DNA can be stored at 4 degrees Celsius for extended periods. However, for long-term storage, minus 20 degrees usually utilized. Avoid any kind of repetitive freeze thawing of DNA since this can cause degradation. And the storage of DNA at 4 degrees is better than minus 20 degree, and storage at room temperature dried with stabilizer is even better. Now, if we talk about organic extraction, what are the pros? It yields relatively pure high molecular weight DNA. DNA is double stranded, which is good for RFLP. Cons will be time consuming, require sample to be transferred to multiple tubes, increases risk of contamination, and it involves the use of hazardous and smelly chemicals. So if we talk about non-organic DNA extraction, it does not use organic reagents such as phenol or chloroform. Digested proteins are removed by salting out with high concentrations of LICL. However, if salts are adequately removed, problems could occur with the RFLP procedure due to alteration of DNA mobility or band shifting. Now, if we talk about cell lysis buffer, we should lyse the cell membranes nuclei with intact pellet nuclei and resuspend nuclei in protein lysis buffer containing a high concentration of proteinase K. Okay, you can lyse nuclear membrane and digest proteins at 65 degrees Celsius for 2 hours, and temperature helps denature proteins and proteinase K auto-digest itself. So to remove proteinaceous material, lithium chloride is added to a final concentration of some 2.5 molar and incubated on ice, and proteins precipitate out and are pelleted by centrifugation. Now, if we talk about non-organic DNA extraction procedure, so DNA remains in solution, transfer supernatant to a new tube. Care must be taken not to take any of the protein pellet. Very careful. DNA is precipitated by the addition of room temperature isopropanol. Lithium chloride will not precipitate with DNA. Precipitated DNA is washed with 70% ethanol, dried under vacuum, and resuspended in TCDTA buffer. So you can see lysis, TCDTA SDS, protein precipitation, sodium acetate, DNA in aqueous solution, DNA precipitation using isopropanol. So if you see, this is an agarose gel that has 5 genomic DNA samples from various plants. So you can note that the DNA at a very high molecular weight and has a clear thick band. So this DNA was extracted in a research lab under optimal conditions. So if you see, it is one thick band, that means the DNA is not degraded. Otherwise it would have been multiple bands or even smeared. So if properly done, genomic extraction should result in bright bands in the very high base pair range of a gel electrophoresis. Why high base pair range? Because these are very big. So this is a ladder. The topmost band will be the biggest band, like the biggest size, and because the smallest band, smallest size DNA will travel faster through the pores, right? So the genomic fragments are almost around 12 kilobase pairs because they are sheared during extraction. So it should have been even bigger than this, but due to shearing of DNA extraction, it's at least 12 kilobase pairs. Now, if you see a smear like this can happen if the preparation is not perfect. That means the DNA started degrading. Okay, so it's broken up into numerous fragments and is not a clean single band at the top. So these are the mid-range size bands, and the right bands at the 100,000 BP range are probably RNA, which also gets extracted during this protocol. So if you have to separate RNA from DNA, you have to be very careful as well. So if you analyze these samples, the first sample is fine, second sample is fine, third, fourth is fine. The fourth sample has severe degradation, can work for PCR but should re-extract. Okay. Fifth sample, sorry. Most of these are shared only, so none of these are completely fine, but the last one is even worse. So coming back to the question that we started with, the aqueous phase in phenol chloroform contains, you're right, it was DNA. So if you have any doubt, I will wait for 5 6 minutes. If you have any doubt, you can ask. Otherwise, after this, we'll move on to the next question. Okay, so we'll move on to the next question for today. So the next question is, which of the following organisms has the smallest genome size? Right? Mycoplasma, algae, crustaceans, and gram-negative bacteria. Anybody?
Mycoplasma. Option.
Okay. So, right, it's mycoplasma. So mycoplasma is it's the smallest and the simplest self-replicating bacteria, right? And mycoplasma cell contains minimum set of organelles essentially for growth and replication, and a plasma membrane, ribosomes, and a genome consisting of a double-stranded circular DNA molecule. So, unlike all other prokaryotes, the mycoplasmas have no cell walls, and they are consequently placed in a separate class called molecules. Like mollis means soft, cute is the skin. So mycoplasmas have been nicknamed as the crabgrass of cell culture. So contamination of cell cultures by mycoplasmas present serious problems in research laboratories and in biotech industries using cell cultures. So the origin of contaminating mycoplasma is in components of cell culture medium, particularly serum or in the flora of technician's mouth, spread by droplet infection. Okay, so difference between mycoplasmas, bacteria, chlamydia, and viruses. So if we talk about the history, the name mycoplasma is derived from the Greek word mycos and plasma. Okay, so basically it was first used by Albert Bernard Frank in 1889. He thought it was a fungus due to fungus-like characteristics. And an older name for mycoplasma was pneumonia-like organisms (PPLO), referring to organisms similar to the causative agent of contagious bovine pneumonia. It was later found that the fungus-like growth pattern of Mycoplasma mycoides is unique to that species. So, in 1898, Knockout and reported that cultivation of the causative agent of CBPP, which was at that time a grave and a widespread disease in cattle herds. So, disease is called by M. mycoides subspecies mycoides. So then this was all his history. So basically, Eaton in 1944 was the first to isolate the causative agent of primary atypical pneumonia in hamsters and cotton rats, and he was able to transmit the infection later to chicks embryos by amniotic inoculation. So because it was filterable, it was considered to be a virus. Okay, but essentially it was shown that to be a mycoplasma and named mycoplasma pneumoniae. So what will be the general characteristics? Very small, can pass through material filters, lack a rigid cell wall, bound by single trilaminar cell membrane that contains a sterol, extremely pleomorphic, varying in shape from cocoid to filamentous to other bizarre forms. Okay, cells are bounded by single membrane showing in section the characteristic trilaminar shape. Cytoplasm contains thin threads representing section chromosome and dark granules representing ribosomes. So mycoplasma, ureaplasma, spiroplasma, and anidoplasma, they cannot synthesize their own cholesterol and require it as a growth factor in culture medium. So acheleplasma synthesizes carotenol as a substitute for cholesterol, but will incorporate cholesterol if it is provided, right? and insensitive to cell wall active antibiotics such as penicillin and cephalosporins. Limited biosynthetic capabilities due to small genome. So that's the issue. So when we are doing cell culture, we use penicillin and streptomycin, pen-strep antibiotics in order to eradicate any kind of bacterial contamination. But these mycoplasmas are very insensitive to these kinds of penicillin antibiotics as well. So even after adding pen-strep antibiotics, your cell culture might still have mycoplasma contamination. So that's why it is a very difficult contamination to deal with. So they can multiply by binary fission, however, cytoplasmic division may lag behind genome division, and this results in the formation of multinucleate filaments and other shapes. They do not possess flagellae or pili. They are non-sporing. Stained poorly with gram stain, can be stained with GMS and DNS methods, and are considered as stable L forms by some researchers, but its hypothesis is still not fully accepted. So their cells can divide by binary fission or first elongate to multinucleate filaments, which subsequently will break up to coccoid bodies. Now, the membrane proteins and glycolipids exposed on the cell surface are the major antigenic determinants of mycoplasma, right? In mycoplasma. So, antisera containing antibodies to these components will inhibit growth and metabolism of the mycoplasma, and in the presence of complement cause lysis of the organisms. So, these properties are used in various serologic tests to differentiate between mycoplasma species and serotypes and detect antibodies to mycoplasma in sera of patients. So some mycoplasmas possess unique attachment organelles which are shaped as a tapered tip in M. pneumoniae and M. genitalium. Mycoplasma pneumoniae is a pathogen of the respiratory tract adhering to the respiratory epithelium primarily through attachment organelle. And interestingly, these two human mycoplasma exhibit gliding motility on liquid covered surfaces. So the tip surface structure always leads, again indicating its importance in attachment. So one of the most useful distinguishing features of mycoplasma is their peculiar fried egg colony like shape, consisting of a central zone of growth embedded in the agar and a peripheral one on the agar surface. So mycoplasma genome is typically prokaryotic, consisting of a circular double-stranded DNA molecule. The mycoplasma genomes are the smallest recorded for any self-reproducing prokaryote. And in the mycoplasmas, the number is estimated at fewer than 500, about one-sixth the number of genes in E. coli. So mycoplasmas accordingly express a small number of cell proteins and lack many enzymatic activities and metabolic pathways. So their nutritional requirements are correspondingly complex and they are dependent on a parasitic mode of life. So if we do taxonomic classification, kingdom is bacteria, phylum Firmicutes, class is molecules, orders will be ontoplasma on ontoplasma tails. Sorry, I am not pronouncing it properly. So you can check out this taxonomic classification in books also. Okay. What are will be the cultural characteristics? So, aerobes and facultative anaerobes except anaeroplasma, which is strictly anaerobic for primary isolation and atmosphere of 95% nitrogen and 5% carbon dioxide is preferred. They can grow within a temperature range of 22 to 41 degrees Celsius. The parasitic species growing optimally at 35 to 37 degrees Celsius. For fermentative organisms, the initial pH of the medium is adjusted to 7.3 to 7.8. For arginine metabolizing organisms, it should be around 7, and for ureaplasmas, ranges of pH should be 6 to 6.5. So dependence of mycoplasmas on their host for many nutrients. It explains the great difficulty of cultivation in the lab. So the complex media for mycoplasma culture contains serum, which provides fatty acids and cholesterol for mycoplasma membrane synthesis. So the requirement of most mycoplasmas for cholesterol is unique amongst prokaryotes, and the consensus is that only a small fraction of mycoplasmas existing in nature will have been cultivated so far. Some of the viable cultivable mycoplasmas, including the human pathogen M. pneumoniae, they grow very slowly, particularly on primary isolation, right? And Mycoplasma genitalium, which is another human pathogen, grows so poorly in vitro that only successful isolations have been achieved. There are ways of culturing that also incubation periods, size, platinum loops, and how they can be subcultured. Basically, this is how the colony morphology looks like. Fried egg appearance. You see the peripheral zone and the central zone. So DN stain is popular because it contains azure 2, which and methylene blue, Na2CO3, benzoic acid, and distilled water. So how it is it done? The plate that will contain suspected mycoplasma can be flooded with DN stain, diluted 1 to 10 in water, and it is then immediately rinsed with distilled water and the stain is removed. And the medium is decolorized by adding 1 ml of 95% ethanol for 1 minute and then removed. So the washing step is repeated second time, rinsed with distilled water and allowed to dry. And the colonies are then observed under low power of a microscope. So mycoplasmas with fried egg colony appearance, they are highly granular and stained with a dark blue center and a light blue periphery. Right, so the agar background appears clear or slightly violet. So mycoplasmas other than M. pneumoniae remain stained, but M. pneumoniae reduces the methylene blue after a period and become colorless. This is how it looks. So glucose and other metabolizable carbohydrates can be used as energy sources by the fermentative mycoplasmas possessing the EMP glycolytic pathway, Emden-Meyer-Pernus. And the oxidative phosphorylation does not occur as an ATP generating mechanism. Okay. So breakdown of arginine by the arginine dehydrogenase pathway has been proposed as a major source of ATP in non-fermentative mycoplasmas, and urea plasmas have a requirement unique among living organisms for urea because they are non-glycolytic and lack the arginine dehydrogenase pathway. It has been suggested that later proven experimentally that ATP is generated through an electrochemical gradient produced by ammonia liberated through the during the intercellular hydrolysis of urea by the organism's urease. So, what are the surface antigens made of glycolipids and proteins? Glycolipid antigens are identified by complement fixation test. Glycolipids with similar antigenic structure have been found in human brain, and protein antigen ELISA, a particular technique useful for the identification of isolates based on the ability of antisera to specifically inhibit the growth of the homologous species on solid media. So they can be destroyed at 45 degrees Celsius in 15 minutes, but they are resistant to lysis by osmotic shock, penicillin, and even cephalosporin. As I was saying, they are very much resistant to these antibiotics. Okay. So most mycoplasmas that infect humans and other animals are surface parasites adhering to the epithelial linings of the respiratory and urogenital tracts. So adherence is firm enough to prevent the elimination of the parasites by mucous secretions of urine, right? And the intimate association between the adhering mycoplasmas and their host cells, they provide an environment in which local concentrations of toxic metabolites excreted by the parasite build up and can cause tissue damage. Okay, so because mycoplasma they also lack cell walls, fusion between the membranes of the parasite and host has been suggested, and some experimental evidence for it also has been obtained. Now, membrane fusion would alter the composition and permeability of the host cell membrane and enable the introduction of the parasites hydrolytic enzymes into the host cell. Okay, and recent studies have indicated the presence in mycoplasmas of antigenic variability systems, and these systems, some of which are already defined in molecular genetic terms, are responsible for rapid changes in surface protein antigens. And the change in the antigenic code of the parasite helps it to escape recognition by the immune mechanisms of the host. Okay. So we'll not go into too much detail of all of this. So coming back to the question that we started off with today, sorry, um, the following organism with the smallest genome size will be mycoplasma. Neither of the others, algae, crustaceans, or gram-negative bacteria are the smallest. Okay. So mycoplasma will be the answer. Anybody has any doubt regarding this question, we can talk now. I'll wait for 5 to 10 minutes and we'll move on to the next question. Okay, so we'll move on to the next question for today. The total mRNA obtained from a particular cell type is known as A. Proteome, B. Transcriptome, C. Genome, D. Nucleoid. So I'll wait for a few minutes. Anybody would like to try?
Option B, transcriptome.
Okay, yeah. So yes, it's transcriptome. So basically, this is the or the omics concept. We'll talk about this now. So what is omics? Omics is basically branches of science known informally as omics. Various disciplines in biology where whose names end in the with the suffix omics, such as proteomics, metabolomics, genomics, lipidomics, all of this. So OM is used to address the objects of study of such fields such as the genome, proteome, and metabolome. And so bioinformaticians and molecular biologists were the first scientists to apply the OM suffix widely. Right? One second. Okay, so genomics, there are different kinds of genomics. Genomics is a study of basically the entire genome of the cell, right? So genome is like DNA. If it's a DNA containing organism, then DNA. If it's, for example, RNA containing virus, RNA is the genome. So then it will be RNA will be the genome. So it can be cognitive genomics, like study of the changes in cognitive processes associated with genetic profiles. It can be comparative genomics, study of the relationship of genome structure and function across different biological species or strains. It can be functional genomics, describing gene and protein functions and interactions of using transcriptomics. It can be metagenomics, study of metagenomes, genetic material recovered directly from environmental samples. It can be neurogenomics, study of genetic influences on the development and function of nervous system. It can be pangenomics, study of the entire collection of genes or genomes found within a given species. It can be personal genomics, branch of genomics that deals with the sequencing and analysis of genome of individuals. So proteomics, epigenomics, lipidomics, transcriptomics, nutrigenomics, glycomics, pharmacogenomics, toxicogenomics, psychogenomics, connectogenomics, foodomics, and all of these are now new terms. So you can read more about this online. There's a lot of journals that talk about omics part. So what is the difference between genes and genetics? Genes will be the basic physical and functional unit of heredity, right? And this made up of DNA. It acts as the instructions to make molecules, particularly proteins. Genetics will be study of heredity or how the characteristics of living organisms are transmitted from one generation to the next via DNA. The substance that comprises genes, basic unit of heredity. Then obviously you cannot say genes and genes. It's basically a pun. Genes are molecules in our body that explain the information hidden in our DNA. So they are a small section of DNA for us, and a chemical that has genetic code for making proteins. Right? It will code for a particular sequence of nucleotides, which will ultimately code for a particular sequence of amino acids with the help of codons, and that will ultimately make proteins. So proteins are the building blocks for almost everything in our body, bones, blood, and muscles, and it is the job of the genes to supervise this protein production. Now, genes are present in almost every cell's nucleus and are made from strands of DNA. And DNA is packaged in chromosomes within the nucleus. Mitochondria also have a small amount of their own DNA. So, red blood cells have no nucleus and no DNA. So DNA encodes all genetic information which is needed to build a cell. It consists of four building blocks: thymine, adenine, guanine, and cytosine. And these individual building blocks are also called nucleotides. A feature of DNA is that these nucleotides are paired. So two pairs exist like AT and GC. So AT and GC, which is adenine, thymine, and GC is guanine, cytosine. So there'll be two there'll be two bonds, hydrogen bonds between adenine and thymine, and three hydrogen bonds between guanine and cytosine. So this is the reason why DNA is double helix basically. So, what is in blood for you to obtain a genomic marker? That is the question. So, what is I've already talked about what is gene. Now, what is genome? Genome encompasses the entire set of genetic information across all 23 chromosome pairs, right? It includes all genes, gene modifying sequences, and all the other stuffs in between. So, genome is basically the complete set, and the genome is the entire DNA content that is presented in one cell of an organism. Genomics is an area within genetics that concerns the sequencing and analysis of an organism's genome. Now, what is functional genomics? It attempts to answer questions about the function of DNA at the levels of genes, RNA, transcripts, and protein products. And a key characteristic of functional genomic studies is their genome wide approach rather than a more traditional gene by gene approach. So, the high throughput analysis of all expressed genes is called transcriptome analysis. So transcriptome analysis can be conducted by two approaches: microarray based approaches and sequence based approaches. Then there will be structural genomics, which seeks to describe the three-dimensional structure of every protein encoded by a given genome. Then we have epigenomics, study of the complete set of epigenetic modifications on the genetic material of the cell known as epigenome. And the epigenome is a multitude of chemical compounds that can tell the genome basically what to do. Then we have metagenomics, study of metagenomes, genetic material recovered directly from environmental samples. And what is comparative genomics? It is a comparison of one genome to another. Transcriptomics will be basically RNA, the mRNA transcripts. Proteomics will be obviously talking about the entire protein set. Metabolomics will be the entire metabolite set. Then we can talk a little bit about DNA microarray experiments as well. So hybridization between two DNA strands, property of complementary nucleic acid sequences to specifically pair with which each other to form hydrogen bonds between complementary nucleotide base pairs. Now gene not active in either normal or disease sample, and gene is active in both normal and disease samples. If you can see over here, this is how a microarray is read: a normal sample, disease sample. If we see this kind of demarcation, that is gene active in normal only or gene active in disease only. You can see, like basically, however, we want to see if this particular gene transcript is upregulated in disease as compared to normal or not. Then we can try to figure out if there is a role of this particular transcript which will ultimately lead to a protein in this particular protein, basically in this case. Then there is all role of mRNA sequencing as well. Now, what will be pharmacogenomics and nutrigenomics? Basically, it's a branch of science that will examine personalized reactions to medications and nutrients. Okay. So, in the future, drug labels may contain information biomarker genomic biomarkers and describe like drug exposure and clinical response, risk for adverse events, genotype, specific dosing, and all of that. Okay. The role of microbiome. The microbiome is defined as the collective genomes of the microbes that live inside and on the human body. We have about 10 times as many microbial cells as human cells. And the gut microbiome plays a very important role in your health by helping control digestion and benefiting your immune system and many other aspects of health. And metabolomics, it's a field of omic sciences that characterizes differing metabolites. It involves evaluating the therapeutic outcomes of clinical drugs by correlating baseline metabolic profiles of patients with their responses. For example, aspirin, 25% of patients who are high risk for atherothrombotic events or aspirin resistant. Metabolomics labeling is being used to diagnose disease, understand disease mechanisms, identify drug targets, and customize drug treatments. So, coming back to the question that we started with, total mRNA obtained from a particular cell type is known as transcriptome. Okay, does anybody have any doubt in this question? Okay, so if you have no question, we can conclude the session for today, and we'll meet again tomorrow. So the last Tuesday I couldn't take session because I was not in India. So the last session would not happen on last Tuesday. So we'll meet again tomorrow from 7 to 9. We'll discuss some more questions so we can conclude the session today, and then we'll meet again tomorrow. Okay. With week 10 of 10th live session. Okay. So we can conclude the session for today. Thank you.
Thank you, ma'am.
Thank you.
Thank you, ma'am.